אחר צהריים טובים. אני שמח להציג את המרצה הפלנרי שלנו לאחר הצהריים, פרופסור אמיר עמדי מאוניברסיטת רייכמן. אמיר משלב ידע בחקר המוח עם פיתוח של טכנולוגיות חדשניות. אני חושב שכתבת חדשות חדשניות נשמע יותר טוב, ויותר נכון גם. כן, חדש או לא בהכרח חדשני. והוא לאורך השנים פיתח כמה דברים מאוד מדהימים, שאני לא יודע על כמה מתוכם הוא יספר לכם, אבל זה בטוח יהיה מאוד מעניין. אמיר, בבקשה.
Okay, תודה רבה. I will switch to English. Feel free to ask questions. This is going to be very informal and kind of an overview of the concepts and the work that we did in my lab and during my career, from the first work in my PhD till unpublished results from the last few months. I want to start with a few take-home messages that I want to hope to manage to get through. And then it's going to be kind of interactive, general public kind of talk. So I hope it will be engaging and maybe even entertaining. So the first take-home message I want to provide in the context of the critical periods theory that won the Nobel Prize in 1981 by Hubel and Wiesel. I would try to suggest a revision to this theory and suggest that representations in the brain is much less sensory. We think about the brain as processing vision, audition, touch, movement, language, memory. And I would try to argue that the representation in the brain is much more abstract than treating it as a unisensory entities that are kind of each brain area dedicated to different functions. I will then offer some data and a major revision to this critical period theory and suggest what we should keep from this theory and what we should revise. And then the results I'm going to share with you suggest that this opens the way for pushing away from homeostatic plasticity. Homeostatic plasticity is basically the tendency of the brain to get to baseline. Yeah. So, and this is responsible, for example, for addiction. Yeah. I'm going to talk a little bit about the reward system, areas like the VTA and the nucleus accumbens. This is the areas that gives us amazing reward if we take cocaine, for example. But then the brain say, oh, the activity is so huge and radical, so let's downregulate, for example, the receptors that are responsible for dopamine or for others. And then we have less and we need more and so on and so forth. So the brain always tries to put, and then if you're abstinence for a period of time, it will go back to the baseline. Yeah? So this is homeostatic plasticity. And in a way, it limits the way we can push the brain homeostatic in the direction we want to, for example, in case of diseases. Yeah, if we have Parkinson, if we have Alzheimer's, if we have depression, I mean, clinical depression and so on and so forth. So I would suggest that the ways to push the brain away from this type of plasticity opens the door for a lot of potential new treatments and new technologies and new ways to treat the brain and the body. And this would be, so the first talk would be more like basic science and try to give you all the data in relation to it and also some technology that we use to address this basic science question. And then in the second half, I'm going to show you a few examples, recent examples of how can we leverage this type of neuroplasticity in medicine. And this would be in the context, for example, of the connection between the brain and the immune system. Some very interesting novel findings there, where I think Israel is really in the forefront of this field at the moment. I would mention in the beginning some work by Professor Michael Schwartz, by Asya Rolls, and so on and so forth, and some work we did in relation to this. And I would show how these new ways to connect different systems and modulate them and change them opens the way for a new way of medicine that I'm going to present in the second part of the talk. Yeah, a little bit of ground support and the team members, in case I have also a longer slide in the end. But I want to mention, besides, the Israel Science Foundation and DOD and Mafat and so on and so forth, the main contribution to the work of our lab comes from the European Union. And the relation, and keeping this relation is super important. So about 4 grants from them, 2 of them ERCs. And really, without them, we couldn't do the work that we present here. And of course, without people that are supporting, I want to especially mention the work of Ofek Salama, the lab manager, Elena Agius-Vela, which is a postdoc from the Italian Institute of Technology, Merav Catalunya, Adis Nir, and Katja Chesla. I'm mainly going to show their work. So let's talk about this critical period theory. What is it? So the critical period theory is based on a series of experiments done in animal models by Hubel and Wiesel. And this, so first of all, let's say 2 very important things about Hubel and Wiesel and about this line of work and other work that gave them the Nobel Prize. First of all, they were very central to show that everything we do in the brain is related to electricity in a very direct way. Yeah? So when we see, when we hear, when we hug, but also when we feel, when our even character and so on and so forth is a result of electric activity. And they were the first to record and show the organization of the visual system electrically to outside stimuli. Okay, so that was one set of experiments they did. And the second set were related to this theory of critical period. And the experiments are amazing. Yeah, they're really super interesting. And what they did was quite simple. Yeah, what they showed is that if they take an animal that otherwise is perfectly developed, so everything is fine with this animal, it's born, and then during the course, if you choose a time window after so in principle they have everything. They have the photoreceptors, the brain areas, everything in their visual system is 100% fine, and they just put a blindfold on one eye for a few weeks, yeah, and then they put a little Moshe Dayan, yeah, on the eye, and then remove it. This eye, which is perfectly normal, the photoreceptor that convert light to electricity, we said already electricity is the language of the brain, are 100% fine, all the pathway, nothing harmed in principle by this procedure. This eye is going to be blind for the rest of the animal life, okay? And if they close both eyes, although again everything is perfectly normal, this animal is going to be completely blind and it's irreversible. Okay? On the other end, if they do the same set of experiment later, yeah, let's say when the animal is equivalent for or not already a baby, a few months baby, then everything is fine. You remove the 2 blindfolds after a month, and a few minutes later, the animal is behaving perfectly well. Okay? So that's led them to the concept 1, that the brain is super plastic when we are young. Yeah? And second, we must receive unisensory information, in this case vision information, to develop the ability to see. Okay? And it's needed in this extra super plasticity period. Okay? And actually in the first part of the talk, I'm going to show some data to support this, yeah, that there is a massive plasticity early on. But on the other hand, the empty part of the half glass, yeah, is that this really limits our ability to make massive changes in the brain to push us away from this homeostatic plasticity if something is wrong, or if we want to create super abilities, yeah, when we are older. It's according to the classical critical period theory, even after the age of a few months, or even 5, 6, the plasticity drops dramatically for these massive changes. And this need to get unisensory information to develop vision, audition, and so on and so forth. Okay? That's clear? I know that you are not coming from neuroscience, some of you, so most of you. So if something is not clear, ask me. Yes?
What is the definition of unisensory? I imagine from the name relating to a single sense, but
Yes. Fantastic. Great that you asked, because I forgot to mention something very important. If you open today, it means basically exactly what you said, that now I'm regretting I didn't have I need to stay here. Anyway, it means, first of all, that there is a division of labor between the different senses and the systems. So unisensory means a separate sense, vision, audition. So all this 30% in the back of our brain, back 30% dedicated to vision, because we are very visual creatures. And a lot of the classic neuroscience came from vision. And then we have 20% of this purple area, audition and touch and movement and language here, and then other systems like memory and so on and so forth. So under this assumption that you must get visual input to develop the visual system and its specializations that I'm going to mention soon, to see faces, to read, or the same with the audition. You need to receive auditory input to develop these abilities, and so on and so forth. And another thing, which your question alluded to, is that it gives us, and this is images that still we open in 2024 in any neuroscience book, medicine book, and so on and so forth, that gives you the impression that actually there is a very sharp borders, and there are almost separate systems that somehow very high, you maybe combine them. So the systems are kind of separate. There is a division of labor principle. And also you must receive each sense input during this critical period to develop the ability normally. Okay? Great. So I want to start with a very brief demo to try and suggest something might be already wrong with this kind of concept. And then I will show you, of course, some experiments and some data. So let's say I'm putting here on the screen two images, two visual images that probably you are not familiar with them too much. And I'm going to randomly assign names to them. Yeah? I'm going to call one of them I would call Kiki, and one of them I'm going to call Buba. Okay? It can be also Didi and Lula. But let's keep it with the Buba and the Kiki. Okay? So in principle, and this is completely random, yeah, I can change the names, I can change the order, and so on and so forth, and I'm going to put you in a spot that you need to make a decision very quickly. Now in 4 seconds, I'm going to point to one of these images, and you need to shout the name that you choose. So you need 4 seconds, and in principle, since you don't have previous familiarity with them, it should be about 50-50 here in the audience, yeah? By statistics, it's not big enough sample, but let's see. Okay? Okay, so take 4 seconds. You have a decision? Great. So you want me to choose the right one or the left one? The right one, okay. 3, 2, 1.
Buba.
Great. Raise your hands. Who said Buba? Yeah, raise your hand. Who said Kiki? So the interesting thing is that if we do this thing, in any language, in any age, in any place in the world, the results are 90% and over would say this is Buba, and this is Kiki, this is Lula, and this is Didi, and so on and so forth. And less than 10% weird people. I have some explanation for this 10% people, but I'm not going to share it because it's less I think, makes sense, and it's less related to the talk. But when I try to explain what's going on here, the explanation that anyone have an idea why you chose Buba and why you chose Kiki?
I at least know what's the intention that caused me to say Kiki Buba.
No, but you are the weird one. I don't want to dedicate time to you. We'll do it in the question and answer. Yes?
Both sound softer.
Yeah. Buba sounds softer and Kiki sounds sharper. What do we mean, sound softer? What do you mean? What is it sound softer?
The change in the voice is more similar to the change.
But yeah, so it's a good hypothesis. So we can do it Buba and Kiki. We can do it monotonously. We can change the order. We can choose different names. It stands still.
You can cut yourself with Kiki, but
you are in the direction, by the way. But yeah.
You can cut yourself with Kiki, but not with Buba.
So it's a very abstract thing to say. Yeah. So here is my explanation. Okay. And again, it's a theory. Anyone can give his own explanation. The explanation I came with, and we will talk about this system later in the talk, it's called the mirror system, is that when you hear booba, your brain automatically simulates saying booba, even if you didn't move your lips. And when you do this, think about booba. Booba involves very round shapes in the mouth, in the sensory motor system, the touch and movement system. And kiki is very sharp movements, yeah, and touch. And the same with Didi or Lula. This is why we can use different variations.
Like vowels and consonants?
It's the actual shape of the motor system and the touch system. Boo-ba, Lula. And kiki is very sharp. And you need to get a decision in uncertainty. And this information goes unconsciously in your system, in your brain. And it's shifting you towards saying booba kiki over 90% of the people. Okay? So this is my explanation. And already with this explanation, we see that all this notion of very separate systems doesn't hold water even for very simple stimuli, like very simple geometrical shapes. Yeah? So this is the goal of this demo. Yes?
Wouldn't the best example be taste and smell would combine together to a degree?
Yeah, and there is many more. There is the McGurk effect, which is actually a very surprising effect. I'm going to give you the sound ba, 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 auditorily. Yeah? And I'm going to play visually pa, va, ba. You are going to hear what you see and not what you hear. Okay? So this is another example. Next time you go to the theater, look at the movie. It looks like the sounds, the voices come out of the mouths of the actors, yeah? But actually, if you close your eyes, you see it's coming from the speakers, and it's called the ventriloquist effect. It shows also the dominance of the visual modality over other modalities in many cases. But yeah, and actually there is even more extreme example for this, and this is synesthesia. About 5% of the people, some would even say 10%, of the population have this phenomena. Billie Eilish, Pharrell Williams are modern examples. Kandinsky is another, in which, for example, certain sounds would trigger visual phenomena that are not existing in the real world, and very consistent, yeah? Another example is people that see letters in colors, yeah? They actually see the color, yeah? Yeah, absolutely. Okay, so this is an extreme example which shows the systems are really connected to each other. Another example, as I said, that we'll get back to it in the second part of the talk, is this notion of separate systems between the brain and the immune system. For many, many years, the notion was that immune cells from the immune system cannot cross the blood-brain barrier, and this is completely separate systems, yeah? And Michal Schwarz's work, for example, showed that the pharma company, and again, it's a theory that's now being tested and the drugs developed, but there is a lot of amazing science behind it that suggests that the pharma companies invested trillions of dollars to try and solve the problem of Alzheimer's by developing drugs that will destroy plaques. The plaques in the brain in Alzheimer's basically kills the neurons. And if you don't have 20 minutes of oxygen or glucose, the metabolism, for over 20 minutes, these cells will die. So this plaque making them die. And drugs were aimed at removing these plaques. And it was a complete failure. There is now one drug that was approved recently. It has a very minor effect if you diagnose very early. And it has terrible side effects. Yeah? So this is the results of trillions of dollars, several decades of research. And what Michal Schwarz said and showed in animal models, and I will show you some data from human, is that it might be that there is an increased inflammation in the immune system, increased inflammation in the brain, maybe they are related to each other, and this starts the cascade of events to lead to this plaque developments and so on and so forth. So it's a direction to show that, and we know today that if we are not in a small stress like me now, giving this lecture, but in a major stress, then for a long period, then we can have increased inflammation, it have detrimental effect. And we know, for example, all the immune system are very affected by our mood and by our mental state and so on and so forth. So there seems to be connection. And even more recent and quite amazing data comes from Asya Roll's lab, where she targeted the key structure in this pathway of the VTA and nucleus accumbens that I talked before in the context of addiction. And what they did was the following. Again, why there is relation between the reward system and the immune system, it's very important. For example, if you eat food, yeah, and there is some viruses or bacteria there, you might get a good reward if it's tasty and good, and if the food is not good, you should not get the reward. And there is interesting interaction people didn't think about the relation of this system that is very central to learning, for example. Yeah, you learn, you need the motivation to learn and get rewards and so on and so forth. And what she did was to target this structure in animal model that have cancer, very controlled way of cancer. And they did something similar to what Elon Musk is doing, yeah. Technically, they did something completely different, yeah. But the idea was to elevate in a dramatic way, it's like a hammer to the system, take the baseline activity of the VTA from here to here, yeah. Take it up. Yeah? And see if it have any influence on the cancer. And the result was there is massive reduction in the tumors following this intervention. Yeah? And there was a proper control and so on and so forth. Another very interesting phenomenon that relates probably to this network is the placebo effect we're all aware of. Yeah? What is the placebo effect? The placebo effect is, say, let's say I have a terrible headache. I'm getting a pill that have water inside, yeah, and I'm feeling better, yeah. So the pharma companies treat this as an artifact. They say, oh, this is a terrible artifact, I believe, and I have some brain activity associated with this belief and this mental state and state of my brain, and this triggers some positive effect, probably through the immune system. And this is an artifact. This is a bad artifact. We need to show that the drugs are better than this. But the other side of this is that since this placebo effect is involving also this reward system and other systems, can we use such effects to optimize the immune system and recovery and so on and so forth? And that's what I'm going to show you in the second part of the talk. Okay? Great. So let's go back to Hubel and Wiesel. So one of the first studies I did during my PhD was to see what happened when you don't get visual input. So from Hubel and Wiesel, we know, okay, we cannot restore vision anymore. But what happened to these people that were born blind and during this critical period received something else? They didn't receive vision. Yeah, they received a lot of other stuff. And there was some studies about audition, about touch. And what we wanted to target is what happened to their memory system. Because we know we had some pilot data that show us that blind people actually have superior memory. Yeah, compensation. A sort of compensation. And what is the neural mechanisms of these compensations? And to make a long story short, we found that one, they have superior memory. So for example, one very quick and intuitive example I can give you to what do we mean memory extension is, if I'm going to give you a random list of numbers or of words, yeah? If I'm going to give you 4 numbers, maybe you are in Adam, so you will remember more. The average person will remember about 7 units, yeah, without studying them. If you study for a prolonged period, maybe you can do more, yeah. And the blind could do much more, like around 13. So they have a much stronger memory. And what we saw, which was very surprising, is that almost half of the 30%, mainly in the left hemisphere, is recruited by tasks that usually are activating the hippocampus, yeah, and the parietal cortex, which are the network that are supposed to delineate memory. And actually, there is a connectivity between V1 and the primary visual cortex. So it's mainly showing a really radical form of plasticity, showing how plastic is the brain early on and taking over this visual function. It's showing that, so you see here that some have more average and some have a little bit superior memory and some have high superior memory. And this is correlated to how much they recruited their primary visual cortex. Yeah? And later we even showed that this is causally related. If we interrupt the activity here, we hamper verb generation, for example, the ability to retrieve a verb for a noun, and so on and so forth. So actually, it's showing that the brain is pluripotent. It's a little bit like the concept of stem cell. If we are early enough, a brain area like V1 that actually encode the dots and lines orientation and map the screen of the outside world is actually now a memory area. And so this fits very nicely with the theory of Hubel and Wiesel. Okay, say, okay, you cannot recover vision, but the brain is so plastic early on that we can change completely the function and rewire brain areas or increase connectivity. And really, I won't go over all the text, but it suggests that human cortices are congenitally pluripotent, and then once you get the outside world, they are like being set apart to vision, audition, and so on and so forth. Okay. Another alternative which we have here that actually I like more now is that actually we do have these connections between vision and audition and touch and memory and language. They are there, but they are relatively weak and suppressed by vision. And when we remove vision, we suddenly can uncover them. We did like a crazy experiment when I was a postdoc and later a faculty at the Harvard Medical School, which we took sighted people and blind them for 5 days. Did exactly what Hubel and Wiesel. There you have a privilege. You can put them in the hospital, have nurses taking care of them. They are out of work, getting a lot of money for this experiment. And we imaged their brain and their abilities, and we summarized it in a review in the plastic human brain cortex. So there is some weak connection that we can uncover using removing this 30% of the visual input. We basically reduce 30% of metabolism, of load of oxygen, and so on, and this might help to strengthen. By the way, at the same time, we show that there is traces for homeostatic plasticity. So I won't get into all the details, but basically what Hubel and Wiesel showed is that we have a retinotopic organization. It means that we have a map, that the center of the visual field is in one spot in the brain, and closer to it, closer, closer. So we have a full map of the visual field of the external world in our V1, V2, V3, what we call the retinotopic area, the area most sensitive to critical period. And we can see traces for this connectivity. So the foveal areas, the center of the visual field in V1 connected to V2, connected to the face area, which likes the center, or reading area, which likes the center, and so on and so forth, even though they didn't get some of these people visual input for 40, 50, 60 years. Okay, so far, we supported this theory. And now I'm going to show you a line of work which used, for the first time, new technology that we developed that aim to see whether we can reverse the picture of this recruitment of memory and others. And the thing that we used for it, so first of all, let's talk about the specialization we have. We already talked about V1 and V2, V6, and so on. I will get back to some of this area later. But we also have what we call another layer of organization, which divide the visual system into two main parts, two main pathways. One goes from V1 to the temporal lobe, which is called the ventral stream. And the ventral stream is interested in what we have out there in the world. Is this a cup? This is a face? This is a chair? What are the things? Yeah? The dorsal stream, on the other hand, is interested in where things are located, especially in the context of movement. So I want to have a sip now. So I need both the shape of this and to open my hand and to know where it's located. So this is the ventral, and then to know where it is and pick it up. That's the dorsal. And within the ventral stream, we have now we have a bit more, but not much more. Roughly speaking, we have about 5 main area specialization for things that are super important for us as human. So the ventral stream would process the shape, of any tool, of any object, but it have a dedicated specializations that, according to Hubel and Wiesel, we must have visual input early on to develop them. And these are, you can guess them, some you can guess, some no. It correlates with some amazing technological inventions we have as a civilization. One of them is tools. Yeah, we are specialized to process tools. Faces, very important for communication, for a lot of other things. I'll talk a little bit about reading and writing, which is actually, from my perspective, the most underrated technological invention in the history of civilization. Yeah? And I will try to talk a little bit about it. We have navigation, which is very important. And the last one is the body besides the face, which is very important for motor learning and many other functions. Yes?
I wanted to ask that according to this theory, then what those areas spatializing in units that, for example, did not have the technology of writing, since we didn't have it until quite recently?
That's a very good question. I will deal with it with data and some theory. Okay, if we have time, I see that we. So I want to, but I will get back exactly to this question in a few slides. Okay. So what, and then, so first of all, the separation to these two streams, you must get visual input, and there is experiment that if we lesion the ventral stream in animal model, they can't do shape recognition, but they can do localization. Classic experiment by Ungeleider and Mishkin from the NIH, and then if we lesion the dorsal stream, we cannot do localization, but we still recognize the shape, okay? And also you can have a lesion here and won't be able to read or to recognize faces and so on and so forth. So this is really functionally relevant. Okay, so what was our plan? Our plan was inspired by dolphins and bats. We say, okay, in the animal kingdom, we have animals that have the human don't have this ability, but they have, during evolution, they developed the ability to see with sounds, okay, to the same resolution as vision, okay? One example is bats. We say blind as a bat. We know this, you know this sentence, yeah? It's a wrong sentence. Blind bats have perfectly working visual system, and they use it. And they have eyes, and they use it. But the evolutionary, they have another trick. If they want to go very deep in a cave, they turn on a sonar system. They emit a lot of sounds at the same time for a window of time, analyze it, and they can trace very, very accurately things. They can discriminate a small cricket from a small stone and eat it and recognize it, and everything is fine. Just from the analyzing the sounds, a lot of sounds together. There is information. It's a little bit like ultrasound also, but with hearing sounds, yeah? So what we said, what was the plan? The plan was to develop a technology that enable us to maintain visual input, the shape, the location, and the color even, in sounds, but not in ultrasounds, not in very high frequency sounds, but in normal hearing sounds, over a window of time. We will have 15 sounds that are in pentatonic scale, and they will draw up the picture in a resolution of up to 1,000, 1,400 pixels. Why we choose this threshold? It's low enough. It's not the millions that we have in the visual system, but it's enough to recognize a face. Okay? Now, I don't have time really to go deep into the algorithm, but I just want to give you the gist of it. But before that, let's understand what was the logic behind this experiment. The idea was, okay, let's go back now to this brain of blind people that was born without retina, or they never saw one photon of light in their life. We know that the visual system are taken by other functions, yeah? And now let's try to show them ventral versus dorsal functions. Let's try to show them tools. Let's let them have read faces and see whether we can restore at the age of 40, 50, 60, the specializations of the visual system using this approach. Okay, it's clear, the theory? Okay, great. Now, how can we do this? I won't be able to really get into all the algorithm, but just to give you the gist of it, let's say we have one sound, and I want to draw a smile, yeah? So you will have to sing with me. I will start with a high note, T, sing. And I will sing with you the first half of the smile, and you continue alone the second half, yeah? I didn't explain to you anything about the algorithm, yeah? Just the high notes denote high pitch. T, T, T. Very good. Now let's do a sad face. Yeah, it starts with low, T. Okay, so this is the first 2 minutes with the blind. Yeah, and we move their hands, and now let's add the second voice, yeah? Usually in music we have 3, 4 voices. In our app we have 15 in the iMusic app, 15 voices, so it's have a lot of resolution. But let's add the second line. You are going to sing in different phase, yeah? And I'm going to add the eyes, okay? You ready? The beep, beep, okay? So again, now we train them for 50 hours, and after 50 hours, you see they can do both ventral stream shape recognition and localization and action. And they can separate different shapes, Sprite bottle, apple, and even differentiate the color. And this is Hamutal. She's congenitally blind. She never saw one photon of light in her life. Yeah? Another example is coming to the home of Sigalit, another congenitally blind. And we put their shoes somewhere, and she explored it with the system, a camera here, headphones here. Okay, now I want to switch gear and show you what happened in the brain. So first of all, when we started their training, most of the activity was localized to this 20% that is audition, fits to the model of separate systems. But actually after training, as early as 10 hours after training, we start getting activation also of the visual system, which correlates with the time they actually recognize the shape. Yeah? So we actually get this 30% of the visual system start responding to this sonification, this like ultrasound in the sound level, all the ventral stream, dorsal stream, and so on and so forth. The second thing was to start and digesting area by area and trying to see if we can get specialization. So the first area we target is called LOC, because we had actually some previous work to this project, which we showed that if we are, actually it was an experiment designed for a completely different purpose. We wondered where there is a location where what pathway in the touch modality. Yeah? Because I can close my eyes, even as a sighted, and palpate things and recognize the shape, and then say this is located here, this is located here, and so on. We wanted to see if there is similar organization. And when we did this experiment, we got a very surprising result. There is some activity in the touch system, but the peak activity that specialized in recognizing the shape was exactly in the ventral visual stream. And then we said, okay, maybe it's imagination, so let's target congenitally blind, people like Hamutal, and see whether they are activated by tactile object recognition without any visual recollections, imagination, and so on. Same result. And then we started to say, okay, maybe there is something wrong in these separate modalities, and maybe there is something wrong in this critical period theory, yeah? And then people, and by the way, another result we got is when we say the names or hear the typical sounds that objects make, there is no activity in the ventral stream. Okay, so this is another part of this riddle, yeah? Then we said, okay, maybe the ventral stream have a much more abstract computation and function. What could be a more abstract computation and function? Maybe it's interested in geometry. It's interested in geometry and shape in the external worlds. Vision is the dominant modality, so it's an illusion, but it seems that it's the only sense or the main sense to do it. But actually, if we give shape and geometry at any age, it could be. And then some of the critics said, you know, maybe the critical period is very long in humans. They palpated objects, the congenitally blind, and they recruited the ventral stream, just like you showed for memory. Yeah. So we said, okay, so then what we want to do is develop this technology that enable us to see objects in different, in sound, in technology they learned in the age of 50, 60, 70, scan their main before and after, and see whether we suddenly get an activation for the auditory modality, because now we have shape in the sound. Before, we have shape only in touch and in vision, but not in sound, because it's association, arbitrary association. And that's exactly the results we got. Okay? And it's published in this Nature Neuroscience work. And then we started to go over other brain area, and here I'm going to the very interesting question of reading. Okay? So first of all, reading. This is my slide of how important reading is, yeah, in our civilization. So we have a kind of evolution. We have, for example, spoken language invented about more than 100,000 years. And this is enough time for evolution to develop two systems, like Broca area, Wernicke area, areas that are specialized in language. But you ask a very good question. Reading language, what's invented around the time of the wheel, is invented only about 10,000 years ago, yeah? This is not enough time in evolution. How come we have this brain area always between the faces and the tool areas in anyone, and it emerges suddenly at the age of 6? Yeah? How can it be? Yeah? Now I want to mention one more thing before we are getting back to it, and what this brain area is doing. And actually, the person that found this brain area, called the visual word form area, notice from the name, visual word form area, that is the main center that is doing reading by vision, discovered together with Professor Laurent Cohen, who was also winner of the Brain Prize. And what he had to come up with explanation. Yeah, how can we have something like this? And his theory was called the recycling theory. What does the recycling theory says? It says the following. Maybe what was going on is that let's try to look what this brain area is doing regardless of reading. Yeah? And what is found out is that this brain area really likes the center of the visual field and not the periphery, which is the high resolution, which is great for reading. And the second, it's area that really like line junctions. So if V1 likes lines and dots, this area is a bit higher, and he likes line junctions. So maybe this was the specialization of this, and this is already there, the structure. And when this was invented, we recycled it. And this is why we see a consistent activation. Okay? And then his postdoc visited Israel. He was a Polish guy, Marcin Swed, did his PhD in the Reichman Institute for some bizarre reason, and he really enjoyed it. And he came to visit in Israel, and he came and presented this work and this theory. And I said, okay, this is a very interesting theory. We already have these results on touch. What do you think will happen if we test people that are congenitally blind, never saw, and they read Braille? Yeah? So, and we started to have, and we had a phone call with Stanislas. He was very open to discuss this. And basically, we had two main options. One is that we will discover the homologue of the visual reading area in the yellow area, in the touch system. Yeah? It makes a lot of sense because we are reading Braille using touch. The other option was that there is something deeper, more abstract, just like it's not about visual objects and line combination and curvatures, it's about geometry. Maybe there is a deeper computation this brain area is doing that is in the basis of this activity, and we will get the same activation of this visual word form area. Okay? It's clear? Great. So we run this experiment, and you can already guess the result. The peak of the group, the congenitally blind group, was exactly in the visual word form area in the left hemisphere. And if we look on the individual subject, we see there is, and remove the group tag, there is no difference between blind from birth reading Braille and sighted reading using text. And then we had to come up with explanation. And the explanation that I came with is to say actually reading is the first cross-modal multisensory technology. It's a very similar technology to what we did with the blind of converting images into sounds. It's actually converting information that is usually part of the language system of auditory modality and converting them into vision, yeah, originally, yeah? So in the beginning of the languages, we had symbols, like the hieroglyph. For every object in the world, there is a different symbol. So now let's say the symbol is for a glass, a cup, yeah? So we need to draw a little cup. And now we have in the visual modality something that before the invention of the writing system, by the way, there is no civilization before the invention of reading. The civilization started, the history, the accumulation of knowledge, and so on, with this invention. This is why I think it's so important. Before that, we were hunting and drawing some images on the walls, and that's it. And communicating, we want to hunt, so we talk auditorily. I talk auditorily, you listen, and we go together and we hunt. But that's basically it, yeah, to a large extent. So we have a visual representation of auditory word, and this is reading. Then people start saying, in parallel places in the world, listen, this is not a very economic way to represent reading, yeah? Because we need so many symbols to learn, yeah? So let's, instead of representing whole auditory words, let's represent phonemes, yeah? A, ba, ga, da, and the I, a, u. And then we have about 30 symbols for each phoneme. And now we have several phonemes. Several phonemes give you a word. Several words give you a sentence. And you have a system to decode this information. And this now, and what the visual word frame area is doing in this case is going back from the symbol into the auditory language words. Yeah? So it's not line junctions. It's not center of the visual field. It's doing something deep that is going back and forth. And indeed, we found a high-end connectivity to this structure. And this is why when we give them to read Braille, we get exactly the same activity, because now the symbols are dots, and each dot have a phoneme, and it's the same function. It's the same computation. Okay? So it's actually enabled us to get a deeper understanding what is the actual representation in the brain. Yeah? And then we said also, this is again, it's against this separation of the senses. So, and people said, you know, but they learn to read Braille at the age of 6 and so on and so forth. So can you try to teach a new text in a new sense that you never experienced during your development and see what is the specialization for this? And we said, okay, now we have a system. So we can have, we ran an experiment in which we gave Roman numerals, V, X, I, L, that have quantity, color, or phoneme. And in each trial, they either did the phoneme, or the quantity, or the color. And we looked what happened in the brains of sighted and with vision and of congenitally blind. And what we found was really interesting is even when you are at the age of 60 and you learn to read, let's say, Hebrew letters for the first time in your life, then you get this specialization in the left hemisphere. As fast as 10 hours after learning at the age of 60. And what, to our surprise, we also found that numbers, there is a number, a human homologue for the reading. It's a number, originally visual numbers, but actually any quantity that is in the right hemisphere. And it's also preserved in the blind. And there is different connectivity. On the left hemisphere, it's connected to phoneme, and the right hemisphere is connected to quantity maps in the parietal cortex. Okay? Okay, the last example I want to give you is the body, because it's kind of a fun, and then we will switch gears and move to the second part of the talk. How can we use this type of brain plasticity? So very quickly, this visual body area is, you know, usually we think that most of the information is in the faces, right? If we are happy, if we are sad, yes, smile, sad, and so on. So what I did, I took 2 soccer players, and I cut only their face. And you need to guess which one of them scored a goal and who missed, based on the face information. Okay, again, 4 seconds to decide. Okay, it doesn't matter. Just choose one, yeah? You ready? Okay, who thinks this guy scored a goal? Raise your hand. Who thinks this guy scored a goal? So about 2/3, third. Here we don't have something very close. It's about half-half. But actually here, I hope it's the only place in the lecture I lied to you. Both of them scored a goal. And if you look at the body, it's very easy to see, and also when you miss. So there is a lot of also social information. But more importantly, this visual body area, notice again the names that were given to this brain area, found by Nancy Kenwisher from MIT. Visual body area is the main feedback into the mirror system. Mirror system is a super important system discovered by Rizzolatti, which have neurons that activate exactly the same. It was found, again, in animal models, in primates. If I'm going to do this movement and take the cup, these neurons would fire. They won't fire for a different movement. But if I'm going to look at you doing this, the same neurons will respond. Yeah? So basic, and what was the goal of this area is enable us to learn. Yeah? If a baby wants to learn how to open this, he would look at me how I'm doing it visually, transform this information into movement, and would do it. Yeah? And indeed, it's very preserved in evolution, this ability. For example, this very cute kitten, it doesn't speak Hebrew or English. Yeah? Okay, and you see it's mapping from vision into movement. Yeah, so this is a very important system that we learn to work. Even humans can learn this. Yeah, as you can see here. Okay, and now let's get back to be serious. What happened in congenital blindness? Yeah? They never had vision, so they weren't supposed to have the ability to convert from vision to motor. Yeah? They never were exposed to visual input. But now we can do this experiment. So we give body shapes and movement to Hamutal and all the other tens of congenitally blind without training. So we just analyze in her brain this shape, weird shape she never saw. And she do it. And we put her in the scanner, and we get exactly the same thing. 100% of the congenitally blind have activity in the visual body area after they learn to extract the shape. So the bottom line is that the representation in the brain is way more abstract, not unisensory. We need a major revision of the critical period theory, and it's opened the door for pushing away from homeostatic plasticity into massive numeric plasticity in the adult brain. So this is work we started in the early 2000s, yeah? And now we can go back into clinics. And for example, this is a recent paper from last year we did, which showed that there are some very rare examples of people born with cataract, and they were born in a village that don't have any hospital there. So nobody treated this cataract. And now a humanitarian effort, in this case it's the Himalayan Cataract Project, come and do the surgery, and we test them and train them after that, and we see that they can learn geometry very, very quickly. Yeah? Another example, also from several papers from last year in our lab, is we develop a technology that enable, I don't have time to show you exactly, this is twins that were born deaf, went through cochlear surgery, had some of their function recovered, but actually there are some functions they cannot do well. For example, speech in a very noisy environment. We found that cochlear implants that are following congenital deafness cannot localize anything in space. So all this dorsal-like stream in the auditory, we know there is whatware also in the auditory modality. They cannot do it all. They're completely at random. And we develop a technology, which I won't get into the details, that this is the work by Adi Snir, Kasia Tesla, mainly in the lab. And we show, first of all, that they are almost a chance. I won't get into zero means they are always pointing in the opposite direction. 1 means they are exactly on the target. And 0.5 means they almost cannot recognize locations in sound. And we give them this touch technology. In less than 1 hour, they can do now very accurately touch, and they can combine it with audition. We remove the touch, the system goes back to baseline. But what I want to stress here is that these twins had 1 year of weekly training on these bilateral cochlear chips and 10 years of use. They can do language in quiet very well. They can have a conversation one-on-one, but they cannot restore the function. And now when we created the technology that is mapping audition into touch, they can do. And now we're seeing if we can teach the cochlear implant to operate without the touch and improve their auditory system. So this is just a few examples. Maybe we can watch a very short clip with these congenital twins just to give this notion of location. By the way, Ben Shani was broughting them, and they were the first cochlear implants, so we were so surprised it's worked. So a sighted can localize. The twin that had the touch system can, with the sound. The twin that don't have the system cannot localize at all. And now they switch. Okay, let's go on. We actually let them enjoy music for the first time, and later we developed something to treat depression and anxiety post-trauma using this, and there is a very interesting story post October 7th, but I won't get into it. Now I want to switch gears into the second part of the talk and try to tackle a few of clinical, it will be shorter, don't worry. I know I have 15 minutes. try to leverage this neuroplasticity in medicine. And I'm going to show you some data in the context of brain neurodegenerative disease, of Parkinson's disease, depression, and so on and so forth. So the idea is the following. We know, and I'm going to do it very quickly, but I gave you all the intro. If we look at Alzheimer's disease, as I said, we have a brain area that we start losing, yeah? And maybe there is some link into inflammation and in the immune system in the brain. So one part of the goal is to say, okay, can we use various technologies like the one we developed for blind and deaf, but also others coming from clinical psychology, coming from occupational therapy, and so on and so forth, that would reduce the inflammation, okay? And we scan their brain before and after and see if there is any improvement in their memory or their other abilities, yeah? The second part of the equation we didn't talk about, there is actually a very nice article from today in Haaretz that talks about the notion that the areas that were the last to develop in evolution are the areas more sensitive to being lost with age in all of us, and it's expedited in people that have subjective cognitive decline or Alzheimer. And this is areas that are the default mode network, that's memory system, hippocampus, parietal cortex. So this is area that involved in memory. What is really interesting is that 2014 Nobel Prize showed that exactly the same network that we use for verbal communication, episodic memory, and so on, is used, like the hippocampus, is used also for navigation. This is the GPS of the brain for spatial awareness and navigation. So we said we not only have interventions that would increase inflammation, and we'll test it by taking their blood, yeah, and testing them clinically, or saliva, we are also going to extra train this spatial navigation system. Actually, it's very easy to say. And this is a bit of the new connection between the brain and the immune system. I won't get into all this. So we developed this maze. Let's see if it's working. Yeah. And again, this is based on a lot of work we did on navigation in the blind with Daniel Shabat. So basically what we did, we took a classic mazes using animal models to studying the memory system called Hebb-Williams maze. We created a game, a very rewarding game, that enabled them to learn, explore visually this. But we did a little trick. The trick was we took an algorithm we developed for blind people to navigate by sound. I'm not getting into the details, but very easy to learn. And they have the sounds together with the visual navigation, yeah, with this game. After they manage the most simple maze, they get it again, but this time we're 50% cloud. So they couldn't see half of the maze. And now they start to need to use audition, like the bats and dolphins, to navigate. And the third stage, there was completely no visual input, and they only navigated with blindfold with this maze. And in this last stage, what we did is using this trick that I told you about this blindfold experiment, we reduced 30% of the activity in the visual system, because there is no visual input, and we had a lot of data in the lab to suggest that this increases the neuroplasticity and the connectivity and the activity in this system. So this is what we did. And we had several trials. I would start with the first. If I won't manage to show you all of the data, this is a summary slide that basically shows the bottom line of these experiments that we did. Yeah. So the first experiment was related to people who suffer with subjective cognitive decline at the age of 60, 65. And what we showed is that following 3 weeks of daily training with this reduction of inflammation and improvement, gamification, rewarding gamification of this, with this blindfolding protocol, we can improve their memory abilities and the spatial awareness. But we also accompanied with brain imaging and resting state fMRI, and we see that actually, if I want to summarize it without too many slides, if I'm going to put your brain in a MRI for 10 minutes without you doing anything, yeah? And I'm going to, using computational methods, track the strength of connections between brain areas. Just like AI, the most important thing in the brain and in learning and in any representation is the strength of connections. Yeah? In AI, we call it the weights in the neural network. Yeah? And in the brain is when one brain area is firing, is the other brain area firing also, or is deactivated, or is completely unrelated to it? So if, and we know there is fluctuation in the brain, so if the brain area is connected, we'll see correlations between them, or some other measure like granular causality for causality, and if not, they would be unrelated. And if I'm going to do this to you, to an average brain, I can know your exact age, or not exact, but there is some statistics involved here and some. But in general, I can tell you exactly what is your brain age if you are healthy, if you don't have any disease. So if you are 65, I can know from these 10 minutes that you are 65. If you have subjective cognitive decline, you are 65, but your brain will look 72. And if you have Alzheimer, your brain will look 80, okay, or 75. And what we see is that after this training, the brain connectivity goes back in time. So it's becoming stronger and healthier and because we lose this connectivity inside this network with age, just like this article in Abetz. The second thing we did was, in the different trials, we took blood samples and analyzed, in the first study together with Michal Schwartz, later together with a Remepy company, so a bit of disclosure. This part is, I have a company I founded, and I'm the chief scientist there, so take it with a grain of salt, because it's also have some business goals to try and bring this new concept of hybrid drugs that I will talk in a second. And we saw that we actually, when we look at the immune system level cytokines, which are inflammatory markers, T cells, and so on and so forth, we look on the immune system, we see we can reduce significantly markers of inflammation. So using this 3 to 6 weeks intervention, we can reduce inflammation. So in a way, we are targeting both sides, both the inflammation and the brain connectivity that's associated with subjective cognitive decline. The second study was related to depression and anxiety, which is a big epidemic in the world at the moment. And the main medicine we have today is SSRIs, yeah, and people take them. When you look at the data, you see that the effect is quite modest, yeah? And it takes several weeks to develop this effect. We measure it, for example, using CESD score, yeah? And this time we develop interventions that are more in the psychological and inflammation domain and less in the navigation, although they did also for this. So I didn't show you a lot of data, but I will show you maybe one slide. What you see here is the individual changes in the memory, spatial memory performance in mazes of the individual with subjective cognitive decline. So some of them don't show any improvement. Some of them show modest improvement, and some of them show quite remarkable improvement. And this is correlated with how much we change exactly the network, the connectivity between the middle temporal area and the frontal area. This is exactly the areas that are middle temporal area, including the hippocampus, most sensitive to die with age and with Alzheimer and subjective cognitive decline. So there is a significant correlation individually by this improvement in memory scores and brain activity. And the same also with the cytokine levels. So here you have changes in IL-18, a very important cytokine that marks inflammation. And here is the connectivity we changed this time in the emotional system. Yeah? And also Precunus also involved in the, some of the area involved in the reward system. Second trial, as I said, was much bigger, had about over 100 subjects. And again, here we target different type of activities. I won't get into all the details, but what we did was actually quite simple. In addition to all the things we did in the first trial, we went on to see which type of therapies worked and had evidence that in clinical trials they can improve depression. Yeah? So for example, cognitive-based therapy. Yeah? It's something that you come to a therapist, it's helping you to do, or mindfulness-based meditation, breathing exercise from specific types, and so on. And what we did was digitize them, making them rewarding, as playful as the game, in addition to all the other intervention we did in the first trial. And what you see here is that we get 27% decrease in their depression score. This is several times better than SSRIs in 3 weeks. And we didn't get it for the placebo app. Yeah, so of course we tested placebo. And what is interesting is that not only we improve depression, reduce anxiety, increase well-being, resilience, stress. These are all clinical scales that were measured independently before and after the trial. After that, we said, okay, but is it maintaining if I'm not doing it daily? So we did another 3 weeks of a follow-up, which we did only 2 times a week. And we continued, not only they went back to baseline, they continued their improvement in all the scales. And again, I would skip this. We see a correlation between how much change they have in their CSD score, and again, the correlation from the amygdala, which is the main emotional center, and other areas in the prefrontal cortex. So again, there is a correlation. And I think the most exciting results we have, I won't show the data, I have 5 minutes, and I want to try and take you to what is the vision for the coming next year. We did a trial on Parkinson's disease, and this was done like the FDA is approving drugs. So it was double-blind, placebo-controlled. So and we basically what we assess is the motor and non-motor functions of Parkinson. Yeah. So Parkinson, they have a big problem to initiate movements, and we know exactly why it's happening. Yeah. Why it's happening? We have cells here in the SNC that have dopamine inside them, and when these neurons are dying, we have less dopamine in the system. It's a very complex pathway in the basal ganglia. Professor Hagai Bergman in Israel is one of the world leaders that characterized this and developed a therapy. This is Merav Cataluna, which was a postdoc in the lab and now works in Remepy, is leading the imaging effort in some of these trials. What happens is that when these neurons are dying, there is different effect on different pathways in the basal ganglia, but the bottom line is that there is an over-inhibition. All of this computation is going into the ventral lateral thalamus, a nucleus in the thalamus, that is actually executing, approving, or inhibiting a planned movement. Yeah? So if there is over-inhibition, this connection is weakened. Yeah? And now we have this freezing. Actually, the tremor is a small side effect. It's socially not pleasant. But the main problem is that I'm walking, and I need to turn right, and I cannot. Or it takes me a lot of time. Or I want to raise this cup, and I'm freezing in over-inhibition. Yeah? And there is a clinical scale to measure it. And the people that measure assess these patients before and after. And again, I won't get into all the details, but it did this maze navigation because it's including fine motor skills and reward and cognition. We do all these things that reduce inflammation and reduce depression and anxiety, and we saw effect on this, and so on and so forth. But what was the most astonishing result is that when we looked after this 3 weeks trial of a daily dosage, we got only in the group that used this app, an increase in this connectivity. So now they are, and this is on top of their medicine, okay? And this improvement they got clinically was correlated with improvement in this exact connection between the ventral lateral thalamus and the motor cortex. Okay? So what I want to, and there is also very encouraging result we got from some of these trials that suggest that what Asya Roll's lab was doing, elevating the activity and the connectivity in the reward system by doing a surgery and implanting molecules and genetics to elevate the electricity, we can do non-invasively and improve elevate the activity level and the connectivity in the reward system. And that's opened the door for intervening almost with any disease that have in a neurological or immunological condition. Yeah? Like neurodegenerative disease, autoimmune disease, and so cancer even, because there is immunotherapy and so on and so forth. So for the last 2 minutes, I want to suggest that a new concept that we are putting forward, and now we are collaborating with several pharma companies, and this is the concept of hybrid drug. And the idea is that we have a molecule that we come from pharma, and we have changing the molecules in an immune system and in the brain and the connectivity, And they work together, and we optimize them together, and we try to go through FDA together and test them clinically just like a drug. And the first trial that we are going to launch, phase 3 trial in the next year, and it's already set up with the international collaboration, would be a double-blind, placebo-controlled, multi-center trial on Parkinson's disease with a combination of levodopa, it's the main drug that's being used to reduce this inhibition. But as you are progressing with the disease, and because of homeostatic plasticity, the effectiveness of the drug is less and less, and they need higher and higher doses, terrible side effects, and at some point, it's not really helping. So if you can slow down the progression, elevate the symptoms, and we have some very interesting pilot data that shows that we can do exactly this. So to summarize, I'm going to go back to my first slides and say that this is, by the way, the people. Mainly want to mention Ofek, Salama, Merav Catalonia we showed, Adi Snir and Kasia. Elena was a very important role of the navigation studies. This is some of my colleagues at Remepy, especially I want to mention Or Shoval and Dr. Michal Tzur that lead the development. And the key messages, I hope I managed to convince you. One, representation in the brain is way more abstract, not unisensory. Major revision to the critical period theory opened the door for pushing away from homeostatic plasticity into massive plasticity even in the adult, old brains. And this might change the face of medicine in the next coming decades. Thank you very much.